Good evening, sir. Now I will be presenting my individual lab assessment. So on the previous lab assessment, our group already loaded our dataset, checked the issues, and plotted histogram, bar plot, and also displayed coefficient and p-value. So now I will presenting my individual lab assessment. First, we use this code to calculate the correlation matrix between CO2 concentration and uptake. The correlation of 0.485 between CO2 concentration and uptake shows a moderate positive relationship. This means that as CO2 concentration increases, CO2 uptake tends to increase, but the connection isn't very strong. Next, this This could calculate the present correlation coefficient between CO2 concentration and CO2 uptake. This gives an idea of relationship between these two variables. A scatter plot is generated with a linear regression line to visualize this relationship. Next, the regression model shows that CO2 uptake is influenced by concentration, plant type, and treatment. Higher concentration increases uptake, while the plant from the Mississippi type and those with chill treatments have lower uptake. All factors are statistically significant with very small p-values. The model explained about 68% of the variation in uptake, and the overall model is highly significant, which is 57.7. The residual standard errors is 6.193, indicating the average difference between actual and predicted update values. Besides that, the VIF value for all predictors are 1, meaning there is no multicollinearity. This shows that the predictors are independent of each other and do not overlap, which is good for model. The Shapiro-Wilk normality test for the residuals of the regression model shows a W statistic of 0.959909 with a p-value 0.009215. Since the p-value is less than 0.05, we reject the null hypothesis and conclude that the residuals do not follow a normal distribution. This suggests that the assumption of normality for the residuals may not hold for this model. Then we create a histogram of residuals. So here is the histogram. Lastly, we extract p-values for individual hypothesis testing. The output shows that the p-values are very, very small, showing that each predictors, which are concentration, plant types, and treatment, is statistically significant in explaining CO2 uptake. This means that these factors plays an important role in predicting CO2 uptake. To conclude, the analysis shows that the moderate positive relationship between CO2 concentration and uptake. The regression model explains 68% of the variation in uptake, with CO2 concentration, plant type, and treatment all being important factors. The model's predictors are independent of each other and the residuals do not follow a normal distribution. Despite this, the small p-value confirms that each factor significantly affects CO2 uptake. So that's all for my individual lab assessment presentation. Thank you.